हेलो एंड वेलकम स्टूडेंट्स वंस अगेन टू दिस आईसीएसई रिवीजन सीरीज जिसमें बायोलॉजी और केमिस्ट्री के दोनों के हम सब्जेक्ट्स के आपको रिवीजन चैप्टर्स दे रहे हैं वेयर वी आर ट्राइंग टू कवर द सिलेबस ऑफ बोथ द सब्जेक्ट्स बायोलॉजी एंड केमिस्ट्री। तो उनके लेक्चर्स ऑलरेडी आ गए हैं। वी हैव ऑलमोस्ट कंप्लीटेड चैप्टर नंबर 1 एंड आई एम डूइंग सेल डिविजन राइट नाउ। सो if you have seen my previous video child, then what is the topic that I have covered in my previous video? The previous video topic related mainly to the mitosis of the cell division. So we were already talking about mitosis and meiosis. We have explained the differences between mitosis as well as meiosis. Yes, and then we said why cell division was necessary, and then after that we then moved on to explain the stages of the cell division. Dismay in the previous lecture we have covered entirely child the interface of the cell division. So I just started with the cell cycle, and in the cell cycle I'm completely done with the interface, wherein I have explained you what G1 phase is about, what the S phase, and what are the major events in the G2 phase. So what did we say? That the G1 phase, it makes the cell grow in size, so for which whatever necessary raw materials are needed, that would be procured or that would be prepared by the cell. Then various proteins would be prepared, RNA would be prepared. Yes, so major events take place of like preparation of basically the RNA and the proteins, and the phenomena or the processes such as transcription and translation, they take place in the G1 phase. Okay, the cell grows in size, and there are certain cells which do not want to, after the growth is attained, they do not want to further continue in the next phase of cell division, which is the S phase, where the duplication of the chromosome, the DNA material occurs. So some cells like our nerve cells and the heart cells of the chordates, they will not participate further in the cell cycle. So they will go into which stage? Do you remember? I said that they go into the G0 stage. So after G1 stage, they enter into the G0 stage, which is the quiescent cell, which is also the cells are living, they are entirely living, they carry on their functions, but they are just not participating in the process of dividing into two cells. So after the G1 stage, the cells which further allow themselves to go or enter into further into the G1 stage, they migrate to the S phase, where what happens, children? It is the duplication of the DNA takes place, and also the duplication of the centrioles also begins here. Okay, then the final RNA, proteins, etc., and the organelles are duplicated, or they are multiplied in the next growth phase of the interphase, which is the G2 phase. So we said that the interphase, which is also called as the resting phase, is actually not is a misnomer word. Resting phase, rather they are doing a lot of work in this interphase, which is also the resting stage. So we completed till there, and we did talk about the significance of, little bit about the significance or what mitotic division is actually all about. And we said that the mitotic division is the division of the nucleus followed by the division of the cytoplasm. So we said that the M phase is actually two things. That's karyokinesis, that is the division of the nucleus, and cytokinesis, that is the division of the cytoplasm. Now this karyokinesis, that is the division of the nucleus, is divided into PMAT. So we are going to take up these events of the karyokinesis, that is the division of the nucleus, in today's class. So beginning with the process of karyokinesis, children. We did also discuss this particular slide yesterday. And this slide says that mitosis is also called as the somatic division. Why, children, it is called as somatic division or equational division? Because it takes place in the normal body cells, and it allows for the exact the formation of the daughter cells to be exact copies of the parent cells. Okay, the number of chromosomes or the genetic material is entirely the same as you see it in the parent cell. So that is why it is also called as the equational division, because the daughter cells are genetically identical to the parent cells. So I'll write it down here that daughter cells are genetically identical to parent cells. So therefore, it is also called as the equational division. The daughter cells are almost Xerox copies of the parent cells. It is largely seen in the cells which are diploid, that is, they have got 2n a number of chromosomes in them. And we do see it happening in the haploid cells in some lower plants and some social insects, and it involves a major reorganization of all the cell components like the nucleus and the cytoplasm. There occurs a major reorganization in them. Why? Because a major churning of all the organelles takes place in this particular division, mitosis division. So that is why we say that it involves major reorganization of all the cell components. So this is what we had done till yesterday, and today we are going to begin to study the first phase of mitosis or karyokinesis. So this is also the karyokinesis. And in karyokinesis or division of nucleus of the cell, we are studying the first phase. So I can say that prophase is the first phase of the karyokinesis or the division of the nucleus. Yes? Now, children, take a look at this structure which you see. What are the things that you are able to see here? Now, take a look at the nucleus. Now, this is the nucleus, children. Inside the nucleus, you can see the genetic material like this. So what can I say then? We can say that the genetic material, which was in the form of chromatin fiber in the interphase stage, has begun to condense, and you can see the shortening of the chromatin fibers, and they are starting to appear as X-shaped chromosomes. So you can clearly see the chromatids. Yes, so can we see the chromatids in this particular diagram? What are chromatids? We know what are chromatids. The arms of the chromosome. So these are the chromatids, which are held by a centromere. Yeah, this is a central circular structure. So can we see chromatids here? Yes, we can see chromatid in this nucleus here. Okay, so then we had also said that in the S phase, in the S phase, we had duplicated the DNA and we had also duplicated the centrioles. So those duplicated centrioles which we had done in the S phase of the interphase, they actually begin to move to the two poles of the cell. So if this is the centrioles, so these duplicated centrioles, they will move to the cell, and if this is suppose the nucleus and this is the centriole. So what are we seeing? That the centrioles have migrated to the two opposite poles. So these are the poles of the cell. So what is the event that will I see in the first phase of the nuclear division? That is called as the prophase. It is rightly called as prophase, children, because pro means early, beginning, sabse pehla wala. Yes. So prophase, in the prophase, the duplicated centrioles, they will begin to move towards the opposite poles or ends of the cell. Okay. Then what is it the next that you are going to see? That chromosomes become distinct, that you are able to clearly make out the chromosomes. Yes, we said that, isn't it? We are clearly able to see the chromosomes. The genetic material is no longer in the form of a chromatin thread. Yes, it is no longer in the form of chromatin thread. It has condensed, it has recoiled, supercoiled, and it has formed chromatids, and we can clearly see the chromosomes. See it in the form of an X-shaped structure called as the chromosomes. And we had already duplicated the genetic material, the DNA content in the S phase. So the chromosomes which are already duplicated as pair chromatids, so we can clearly see the chromatids. So if the question comes, when can you see the chromatids? Then we will say the chromatids can be seen very clearly in the prophase. Yes? Then what else happens here? We know all these things. The point number 4, we are already aware of, children, that the sister chromatids, they are attached to each other at a small region called centromere. So this is the centromere, isn't it? And these are the sister chromatids, isn't it? We have studied everything in our previous lectures, what chromatids are, what are sister chromatids, what are homologous chromosomes, what is the state of the genetic material in the interphase, and when does the chromosomes begin to appear, and how are chromosomes formed, and what are they made up of. All that we have seen in our in our previous classes. Children, we can go back and revisit those lectures to get a clarity, and more clarity. They have been explained beautifully with very, very beautiful diagrams, the differences between chromosomes, chromatids, and so on. So the sister chromatids, we can see the chromosomes, we can see the sister chromatids, which are held together by a central region, which is called as the centromere. Then the next major thing that you are able to see is that the spindle fibers appear between the daughter centrioles. Now take a look at the diagram that I have drawn. Now these are the centrioles which have migrated to the opposite poles of the cell. Now, this is the cell and the centrioles have moved to the opposite ends. Now from the centrioles, now these are what, children? These are aster rays, a kind of microtubules which contain the protein tubulin, from which some thread-like structure will arise, which are called as spindle fiber. These aster rays, which are microtubules made up of protein tubulin, they will give rise to some thread-like structures which we call as spindle fibers. So, यहाँ से, children, some thread-like structures will begin to appear. Okay, let me give a different color to this thread-like structure. Let me make it green. So from here, some thread-like structures are appearing. So some thread-like structures are coming out from these aster rays here. Okay. Thread-like structures are coming out from the centrioles and they are going towards the chromosomes. Where are they heading towards? From the poles, some dhage, thread-like structures arising from where? Arising from the aster rays or the microtubules which are present around the centrioles. Wahan se kuch threads, they appear and they, from both the corners of the cell, that is both, because one centriole is at one end, the other centriole is at the other end. So from both ends, you see the threads, they start to come towards the center of the cell. Okay? They are actually migrating towards the chromosomes. Okay? So this is the major event, child, which you see here, that spindle fibers arising out from the centrioles, they start to arise and they begin to move towards, from the poles towards the center of the cell. Okay. Then next important thing is that, child, we are able, now since these spindle fibers, they are migrating from the poles, that is from the centrioles which are located outside the cell towards the pole, they are migrating towards the central of the cell, towards the chromosomes. So eventually they will go and they will get themselves attached to a region on the chromosomes. Yes. So for that purpose, what is needed is that they need to have space to go around the chromosome. That is, they need to go through the nuclear membrane. They need to enter through the nuclear membrane. So they need to go inside this nucleus through the nuclear membrane. Yes. So we will begin to see the disintegration of the nuclear membrane also taking place in the prophase. Okay. We will begin to see the disintegration, children. Very, very important feature here. The disintegration of the nuclear membrane you will begin to see in the first part of the nuclear division. Why? Because the spindle fibers, those thread-like structures, beginning to appear from the centrioles, which are a cell organelle of an animal cell. Now here you may question that the centriole, which is a characteristic organelle of only an animal cell, not found in plant cells. So then what happens in the prophase of a plant cell? Well, in the prophase of the plant cell also, spindle fibers begin to appear from the region outside the nuclear membrane. Nuclear membrane के बाहर से, you will see that spindle fibers begin to appear even without centrioles being present there. वहाँ centrioles तो नहीं है. In the plant cells, you will not have centrioles, but even then, from outside the nuclear membrane, spindle fibers will begin to be formed and they will start migrating and moving towards the center of the nucleus. वो क्रोमोसोम की तरफ बढ़ते हुए plant cell में भी spindle fibers निकलने शुरू हो जाएंगे and they will begin to migrate towards the chromosomes. Okay. Now, in spite of not having centrioles, even then the spindle fibers, they begin to appear and they begin to start moving towards the chromosomes. ठीक है, जिसके लिए वहां भी in the plant cells also you will see that the nuclear membrane, it begins to disappear or disintegrate. Then what else do we see in the nucleus? There is another important organelle called as the nucleolus. What is the function of nucleolus? Children, for ICSE students, the nucleolus, function of nucleolus is important, children. It has been asked many times in your examination. What is the function of nucleolus? Its function is in the ribosome synthesis, which is in turn related to the synthesis of proteins. Yes. So another important highlight of the karyokinesis or the nuclear division is that proteins are not prepared now. All the proteins that were required have been prepared in the interphase, in the G1 and the G2 phases. The proteins, etc., have been prepared. So we are not preparing proteins in the karyokinesis, the prophase of karyokinesis. So for the proteins to be not prepared, actually the organelle which is responsible, what is the function of ribosome, children? The function of ribosome is protein synthesis. और ribosome को कौन बनाता है? Nucleolus, which is present inside the nucleus. So ribosomes which are formed from nucleolus, वो ही नहीं बनेगा. The ribosome itself will not be formed. Why? Because the nucleolus itself disappears in the prophase. ठीक है? Clear? So, striking features, children, you will say that what are the things that disappear? अब ये कैसा स्टार बन गया है? Okay, children, nucleolus, nuclear membranes, they disappear. Spindle fibers begin to appear, and there are other things which also disappear. All the membrane-bound organelles. What are the membrane-bound organelles, children? We have studied in detail about of the cell, जिसमें कि हमने एंडोप्लाज्मिक रेटिकुलम, गोल्जी अपैरेटस, लाइसोसोम्स, वैक्यूल्स, ये सब हमने सारे एंडो मेम्ब्रेन सिस्टम या मेम्ब्रेन बाउंड ऑर्गेनल्स हमने बहुत डिटेल में स्टडी किया है. यू कैन गो बैक एंड वॉच द प्रीवियस लेक्चर्स ऑन दैट. तो ये जितने भी मेम्ब्रेन बाउंड ऑर्गेनल्स हैं, चिल्ड्रन, व्हाट आर द मेम्ब्रेन बाउंड ऑर्गेनल्स? I will write down endoplasmic reticulum, Golgi apparatus, ये lysosomes, vacuoles होंगे, ये सब disappear हो जाएंगे in the prophase. All these membrane-bound organelles, they will disappear in the prophase of the karyokinesis or the mitotic division. तो क्या-क्या गायब हो रहा है? Endoplasmic reticulum, Golgi apparatus, then nuclear membrane is disappearing and nucleolus are disappearing. What is appearing? The centrioles which are duplicated in the S phase of the interphase, they have appeared towards the poles. वो poles पे आ गए हैं और वहां पे उनके पास से एस्टर रेज के थ्रू स्पिंडल फाइबर्स आर बीइंग जनरेटेड. वो धागे जिन्हें हम स्पिंडल फाइबर का नाम दे रहे हैं, वो धागे निकलते हैं एंड दोज धागे, वो जो धागे हैं, थ्रेड्स हैं, दे बिगिन टू मूव फ्रॉम द पोल टुवर्ड्स द सेंटर ऑफ द सेल, रादर टुवर्ड्स द क्रोमोसोम्स की तरफ वो मूव करते हुए नजर आते हैं. हमें बहुत क्लियरली अब क्रोमैटिन फाइबर का ऑलमोस्ट डिसइंटीग्रेशन हो गया है और हम क्रोमोसोम्स बहुत अच्छे से देख पा रहे हैं. क्रोमैटिड्स जिनके जो आर्म्स होते हैं वो भी हम बहुत क्लियरली वी आर एबल टू सी द क्रोमैटिड्स in the cell. So these are the very very important features of the prophase, children. ठीक है, तो हमने क्या किया कि स्पिंडल फाइबर्स आने शुरू हो गए हैं और बाकी न्यूक्लियर मेम्ब्रेन, न्यूक्लियोलस, एंडोप्लाज्मिक रेटिकुलम, गोल्जी बॉडी, ये सब डिसअपियर. तो ये सब डिसअपियर हो गए हैं। तो इसका मतलब क्या हुआ कि जो क्रोमोसोम आपको दिख रहा है, एक्स शेप्ड क्रोमोसोम जो दिख रहा है, वो अब साइटोप्लाज्म में आ गया। Yes, because since the nuclear membrane has disappeared, तो nucleoplasm and cytoplasm have become one, एक हो गए हैं वो, ठीक है। तो पूरा क्रोमोसोम, the entire chromosomes, they have come towards in the cytoplasm of the cell, और वो उन साइटोप्लाज्म में स्पिंडल फाइबर्स जो कि पोल्स की तरफ से, these blue threads which children you can see it in this particular diagram, these blue threads, what are they are the spindle fibers। और यहाँ ये प्लांट सेल है, इसमें भी ये देखो तुम्हारा स्पिंडल फाइबर्स हैं। तो स्पिंडल फाइबर्स प्लांट सेल में भी तुम्हारा बाहर से, बाहर ऑफ द न्यूक्लियस, फ्रॉम आउटसाइड द न्यूक्लियस, सम थ्रेड्स दे आर फॉर्मिंग एंड दे आर मूविंग टुवर्ड्स द क्रोमोसोम्स। वो क्रोमोसोम्स की तरफ मूव कर रहे हैं। क्रोमोसोम के किस हिस्से की तरफ मूव कर रहे हैं? क्रोमोसोम के सेंट्रोमियर की तरफ मूव कर रहे हैं। तो अगर मैं ये दो पॉइंट ले लूं, ये है तुम्हारा स्पिंडल फाइबर चाइल्ड, if this is the spindle fiber that I am drawing, yes, and if suppose this is my cell, yes, तो ये क्रोमोसोम्स की तरफ आ रहे हैं। क्रोमोसोम्स के किस हिस्से की तरफ आ रहे हैं बच्चों? क्रोमोसोम्स के किस हिस्से की तरफ आ रहे हैं? क्रोमोसोम के सेंट्रोमियर की तरफ, ठीक है। What is centromere? Do we know what is centromere, children? Do we know what is centromere? Centromere is the circular region which holds the arms, which holds the chromatids together. Okay. If I draw this as the centromere, तो centromere की तरफ से ये जो, तो ये जो क्रोमोसोम्स स्पिंडल फाइबर हैं वो तुम्हारे सेंट्रोमियर्स की तरफ आ रहे हैं। कैन वी सी दैट? यस, वी कैन सी। तो हर सेंट्रोमियर की तरफ हम देख रहे हैं कि दो स्पिंडल फाइबर, टू स्पिंडल फाइबर्स are migrating towards the centromere of the chromosomes. Centromere of the chromosomes. Can you see it in this diagram, children? Yes, you can see it in this particular diagram. So let us see what is written here. That each chromosome gets attached to the spindle by its centromere. ठीक है, हम सारे स्पिंडल फाइबर्स को सेंट्रोमियर की तरफ बढ़ते हुए पा रहे हैं, मगर डू दे एक्चुअली कम नियर द सेंट्रोमियर? No, they do not actually come near the centromere. They do not go and attach themselves to the centromere. Rather, they get attached to another structure which is called as kinetochore. Kinetochore, which are proteinaceous structures जो कि centromere के पास ही पाए जाते हैं. So there are some, I'll draw it with a different color. These are some proteinaceous structures which are present near the centromere, where the spindle fibers go and get themselves attached. ठीक है, where the spindle fibers go and get themselves attached. So what is this? This is the kinetochore. So what is this kinetochore? It is a protein structure which is found near the centromere. Clearly you can see these red dots which I have made. These are proteinaceous structures which are found near the centromere, through which the spindle fibers will go and get attached. Okay? So the next point states that the nuclear membrane, usually what we do is that the last point of the first stage of karyokinesis, which is prophase, what did we say as the last point? That the disintegration of the nuclear membrane is taking place. Yes, so that will become usually the first points here, that a complete disintegration of the nuclear membrane, you will see it in the metaphase, which is the next stage. Yes, so complete breakdown of the nuclear membrane occurs in this metaphase. And what you see is that, have I drawn chromosomes in a line? Can you see? यहाँ पे देखो chromosomes लाइन में आ रहे हैं, और मेटाफेज पूरी तरह से जब मेटाफेज के स्टेज में हम आ गए हैं, then you will see that all the chromosomes have aligned themselves in the center of the cell. Sorry, children. So all the chromosomes have aligned themselves in the center of the cell. So this is another important feature. The सारे chromosome जो हैं वो center of the cell पे आ गए हैं और एक line में आ गए हैं. ठीक है? यहाँ पे देखो 4 chromosomes have been shown and all those 4, suppose the cell is having only 4 chromosomes, या फिर better clarity के लिए भी हम number of chromosomes थोड़ा कम लेते हैं diagram में, because in human cells में obviously it is not possible for us to draw 46 chromosomes, isn't it? Just to show you pictorially, so we, for simplicity, we take fewer number of chromosomes and try to show you the different stages of mitosis. तो इसमें हमने 4 chromosome की संख्या ली है और वो चारों chromosome आपको सेंटर में आते हुए नजर आ रहे हैं और यहाँ आपको कैन यू सी दोस ग्रीन डॉट्स चिल्ड्रन हियर इन दिस पर्टिकुलर डायग्राम. सो दिस इज द न्यूक्लियर मेम्ब्रेन जो कि आपको यहाँ दिख रहा है और ये सेकंड डायग्राम में मींस यू कैन सी हियर लिटिल बिट ऑफ द न्यूक्लियर मेम्ब्रेन is still visible in the beginning of the metaphase, but here in this stage when the cell has actually entered completely into metaphase, तो हम देखते हैं कि न्यूक्लियर मेम्ब्रेन पूरी तरह से गायब हो गया है. सो न्यूक्लियर मेम्ब्रेन completely disappears. Okay, so it completely disappears in the metaphase. The chromosomes are appearing themselves in the form of a line in the center of the cell, जिसको कि हम equator भी कहते हैं. So the center, on the equator of the cell, so this part, I will say that maybe this is the equator of the cell or center. Center of the cell पे all the chromosomes have come and aligned themselves, and the spindle fibers आपको क्या दिख रहे हैं? They have almost reached the chromosomes and they have attached themselves to the kinetochores of the centromere. Kinetochores which are present near the centromeres in this particular diagram that I have drawn. ठीक है? यहाँ पे आपको red color के kinetochores से all the spindle fibers have attached themselves. Okay? So next point. A small disc-like protein structure called kinetochore is formed at the surface of the centromere. जो centromere के surface पे इस diagram में जो red जैसा structure है, this is kinetochore. ये आ गया है जिससे कि spindle fibers have got themselves arranged. ठीक है? And each sister chromatid is attached to a spindle fiber originating from the opposite poles. Yes, I have shown in this diagram, in my diagram that I have drawn, कि हर kinetochore जो कि centromere पे है उससे दो spindle fibers have come from the poles and they have got themselves attached to. So these were the major events of the metaphase, children. Okay, these were the major events of metaphase. हम start कहाँ से करेंगे? वहां से करेंगे जहां पे हमने प्रोफेज को छोड़ा था. हमने प्रोफेज को कहाँ पे छोड़ा था? क्या कह के छोड़ा था? हमने बोला प्रोफेज में दैट मुझे अब क्रोमैटिन फाइबर नहीं, अब मुझे क्रोमोसोम्स, दैट इज कंडेंस्ड क्रोमैटिन फाइबर इन द फॉर्म ऑफ क्रोमोसोम्स एंड क्रोमैटिड्स आल्सो आई कैन सी क्लियरली. Yes, so I will say in the metaphase that yes, I can clearly see the chromosomes and the shape and the size of the chromosomes are very clearly visible in the metaphase. We started to say in the prophase, but the exact complete shape and size we can actually make out only in the metaphase. तो मेटाफेज में हम क्या बोलेंगे कि shape and size of the chromosomes are clearly visible. The shape and size of the chromosomes are clearly visible in the metaphase. What did we say in prophase? We said that I am able to locate chromosomes now. I am able to see chromatids now because the chromatin fiber has begun to condense. क्यों ये अभी ही शुरू हुआ? क्योंकि न्यूक्लियस डिवाइड कर रहा है. तो न्यूक्लियस डिवाइड करने के लिए वी आर इन द प्रोसेस ऑफ डिवाइडिंग द जेनेटिक मटेरियल इनटू टू हाफ्स. सो फॉर द जेनेटिक मटेरियल जो कि कॉइल्ड पूरी तरह से एक उलझा हुआ धागा है, क्रोमैटिन फाइबर नेटवर्क है, सो दैट नीड्स टू बी डिवाइडेड इनटू टू. तो उसके लिए उसको प्रॉपरली कॉइल होना है, उसको सुलझना है. इट हैज टू कॉइल इटसेल्फ प्रॉपरली. So when it does so, it forms chromosomes, and the actual shape and size of the chromosomes we are actually able to see very clearly only in the metaphase. But the starting that yes, chromatin fibers have begun to condense and chromatids visible जैसे होने लगे हैं, that happens in the prophase. Okay, so depending upon what the question is, you will give the answer related to the appearance of the chromatids, whether it is prophase or मेटाफेज. अगर अपीयरेंस पूछ रहा है तो आप प्रोफेज बोलेंगे. अगर शेप और साइज क्लियरली पूछ रहा है तो यू विल से दैट इन द मेटाफेज यू आर एबल टू क्लियरली सी द शेप एंड साइज ऑफ द chromosomes. Then what is the next thing that we will talk about metaphase, children? हमने प्रोफेज में छोड़ा था कि न्यूक्लियर मेम्ब्रेन begins to disappear. The complete disintegration will take place in the metaphase. And then what will happen? We said that the spindle centrioles जो दो poles पे चले गए हैं, they are giving some spindle fibers from them, which is going from the pole towards the center of the cell, towards the chromosomes. तो वो जो धागे हैं spindle fibers हैं, वो are going and attaching themselves to which region? The name of that region is kinetochore. कहाँ होती है वो? On the centromere of the chromosomes. ठीक है? Then what happens? Each sister chromatid is attached to the spindle fiber originating from the opposite poles. एक सारे chromosomes जितने भी होंगे सेल के अंदर, they will all arrange themselves towards the center of the cell. ठीक है? सब अपने आप को सेंटर ऑफ द सेल पे अरेंज करेंगे. सेंटर पे क्यों अरेंज करेंगे? क्योंकि उनको दो बराबर के हिस्सों में टूटना है. तो वो सेंटर से डिवीजन होगा तभी जाके दो बराबर हिस्से बनेंगे. ठीक है? So they will go arrange themselves in the center of the cell, which and a kind of a towards the equator, जिसे भी हम बोलते हैं, सेंटर को इक्वेटर भी कहते हैं. तो एक इक्वेटोरियल प्लेट का फॉर्मेशन होता है in the metaphase. So what will I write? An equatorial plate is formed. So you can get questions related to any of these events. वो पूछ सकते हैं. They can say that equatorial plate is formed when spindle fibers go and get themselves attached to the kinetochore of the centromere. When? We will say metaphase. The spindle fibers begin to appear, we will say prophase. The nucleolus disappears, we will say prophase. The nuclear membrane begins to disappear, we will say prophase. Completely disappears, we will say metaphase. Okay, the chromatid shape and size of the chromosomes clearly visible, metaphase. Begin to appear as chromatids and chromosomes, prophase. Okay, so you will get questions like this, or they might give you this kind of a diagram. They may give you this diagram or the diagram of a prophase, and they may ask you which stage are we referring to and what are the events of that particular stage. Okay, so you can get it as a long question with a diagram given and questions based on the diagram. That is, any particular stage of the cell can be given pro, meta, ana, and you might be asked questions related to that. Now moving on to the next stage. अब जब सारे chromosomes center पे आ गए थे, मैं फिर से बनाती हूँ. So I am drawing the spindle fiber and I am drawing the chromosomes. Here. Okay. And this was our cell. Okay. So nucleolus पूरी तरह से खत्म हो गया है. These are the 4 chromosomes that I have drawn. Let me draw the kinetochore, जहाँ से spindle fibers have got themselves attached to. So now what will happen is that कि पहले spindle fiber बन रहे थे, they were moving towards the center. Now what will happen is that spindle fibers will shorten. वो अब खींचेंगे उल्टे डायरेक्शन में. They will begin to shorten. They will begin to shorten. When तो ये धागे जब उल्टे डायरेक्शन में मूव करेंगे, when they will begin to shorten, then what will happen? वो chromosomes को बीच से फाड़ देंगे. They will break the chromosome from here. Chromosomes को बीच से तोड़ दिया जाएगा. From this particular stage, यहाँ से अगर मैं ऐसे individual draw करूं, and suppose I draw one spindle fiber, and if this were to move in this direction, if this thread is were to move in this direction, then what will happen? What will happen? It will be like this. ऐसा हो जाएगा structure. Yes, that will be the structure. Chromosomes जो हैं, क्रोमैटिड्स दो हिस्से में बट जाएंगे, दे विल गेट बाइफकेटेड, और वो हट जाएंगे बीच से. Okay, so that is what will happen in anaphase. और ये जो phase है anaphase, that is the sorry children, this is the shortest phase of the karyokinesis. Okay, this is the shortest phase. सेंट्रोमियर जो कि अटैच कर रही है दो क्रोमैटिड्स को, वहां से डिवीजन हो जाएगा. सो कहां से हमने डिवीजन किया, ये कौन सी जगह है, इसको सेंट्रोमियर कहते हैं. So the chromatids are separating. What is separating? Chromatids are separating. क्यों? क्योंकि the spindle fibers are shortening in length now. Okay. The two sister, read the third point, children. The two sister chromatids of each chromosome that separates and are drawn apart towards the opposite poles, pulled by the shortening of the spindle fiber. So the major event of anaphase is what? Number one, shortest phase. The centromere के पास से chromatids फट गया. The chromatids have divided from the centromere and they have moved towards the opposite poles. Why? Because of the shortening of the spindle fibers. Remember this line, very, very important. Because of the shortening of the spindle fibers, what happens is that the chromosome breaks, the chromatids separate from the centromere और वो दोनों poles की तरफ चले जाते हैं. ओके. तो अगर मैं डायग्राम बनाऊं चिल्ड्रन, इफ आई वर टू ड्रॉ द डायग्राम ऑफ एनाफेज इस तरह से, अभी मैं नेक्स्ट स्टेज का स्लाइड आपको शो कर रही हूँ, टीलोफेज. व्हाट हैज हैपेंड हियर इज दैट, what has happened here is that I am drawing the separated chromosomes. Okay. तो ये जो सेपरेटेड क्रोमोसोम्स आप जो देख रहे हैं चिल्ड्रन, विच यू आर सीइंग, दे हैव, वो बिल्कुल पोल्स की तरफ आ गए हैं. Okay, they have come towards the poles. They have come towards the poles. और everything that we said in the prophase, करीब करीब उसका ठीक opposite we are going to say in the last stage of karyokinesis, which is telophase. Okay, everything that we said in the prophase, what did we say? We said that हमें क्रोमैटिन फाइबर जो धागे जैसे हैं, जो बिल्कुल बिखरे हुए धागे जैसे हैं, दे आर बिगिनिंग टू शॉर्टन एंड वी आर बिगिनिंग टू सी क्रोमोसोम्स, हमें क्रोमैटिड्स नजर आ रहे हैं. तो हम क्या बोलेंगे यहाँ पे कि अभी क्रोमोसोम्स जो opposite poles पे चले गए हैं, two sets of daughter chromosomes. Read the first point, children. Two sets of daughter chromosomes which have reached the opposite poles. वो chromosomes जो बिल्कुल super coiled stage में है, वो अब खुलने शुरू हो गए हैं and chromosomes are beginning to lose their identity. So we will say that chromosomes are beginning to lose their identity. The chromosomes are beginning to lose their identity. Let us read the point. Each chromatid or the daughter chromosome which lengthens, it becomes thinner. अब वो thin होता जा रहा है और फिर से जो है वो क्रोमैटिन थ्रेड के फॉर्म में उल्टा हो रहा है. मतलब प्रोफेज में क्रोमैटिन फाइबर क्रोमोसोम्स बन रहे थे सुपर कोइलिंग की वजह से, होने की वजह से, और टेलोफेज में सुपर कोइल्ड क्रोमोसोम्स है वो खुल रहे हैं. They are losing their identity and they are once again getting converted into chromatin fibers. So that is why I wrote chromatin chromosomes are beginning to lose their identity and chromatin fiber is beginning to appear. फिर हमने क्या बोला था प्रोफेज में कि स्पिंडल फाइबर्स बिगिन टू अपीयर फ्रॉम द टू पोल्स ऑफ द डुप्लीकेटेड सेंट्रियोल जो कि दो पोल्स पे चले गए हैं. तो वी विल से दैट स्पिंडल फाइबर्स डिसअपीयर. देखो बिल्कुल बिल्कुल उल्टी उल्टी चीजें, एग्जैक्ट ऑपोजिट ऑफ व्हाट वी सेड इन प्रोफेज, वी आर गोइंग टू राइट हियर इन द telophase. So spindle fibers begin to disappear. ठीक है. Chromatids thin out in the form of chromatin fibers. Chromatids या chromosomes जो हैं वो अब खुल जाएंगे and they will once again become chromatin network or reticulum or fiber. ठीक है. We said in the prophase that nuclear membrane गायब हो गया था, so that the dissolution of the nuclear membrane allowed for the space for the chromosomes and the spindle fibers to move freely within the cell. Okay, so that they could get themselves attached to the kinetochore of the centromere और फिर एनाफेज में वो दो हिस्से में क्रोमैटिड्स को फाड़ दे सकता था. So इसके लिए जगह की जरूरत थी, फ्री मूवमेंट की जरूरत थी, जिसको अलाउ करने के लिए the nuclear membrane had disappeared in the prophase. Now when that purpose of has been achieved, जिसके लिए न्यूक्लियर मेम्ब्रेन डिसअपियर किया गया था, we have done the movement and now we have divided the chromosomes into two parts. chromatids को दो टुकड़ों में मैंने बांट दिया है. And children, after chromatids have been separated here, तो यहाँ भी आपको 4 chromosomes दिख रहे हैं, and here also you can see 4 chromosomes. The number of chromosome has not changed. Okay, because still the number of centromere is the same. We said that count the number of chromosomes by the number of centromere. If the number of centromere is still showing 4, then it is 4 chromosomes. Okay. So the purpose of the dissolution of the nuclear membrane has been achieved by the free movement of the spindle fibers हो चुका, जो कि allow किया to go and get themselves attached to the centromere, जहां से उन्होंने chromatids को दो टुकड़ों में बांट दिया, and the genetic material the duplicated genetic material of the S phase now has been divided into two, और जो अलग अलग opposite corners पे poles of the cell पे नजर आ रहे हैं in this particular diagram. Okay, so then what will happen? So जब वो purpose of the nucleus membrane का dissolution का solve हो गया, तो what will happen in telophase? The nuclear membrane will begin to appear. So the nuclear membrane will begin to appear in the telophase. ठीक है. दूसरा क्या आएगा? न्यूक्लियोलाई, जो कि न्यूक्लियोलाई इज प्लूरल, न्यूक्लियोलस जो कि भी डिसअपियर हो गया था इन द प्रोफेज, दैट विल आल्सो अपीयर, और बाकी जो मेम्ब्रेन बाउंड ऑर्गेनल्स है वो भी आपको नजर आने शुरू हो जाएंगे इन द टीलोफेज. Okay, so the nuclei, the nucleolus, the endoplasmic reticulum, everything will reappear in the telophase, that is the last phase of karyokinesis. अब karyokinesis के बाद, that is division of nucleus is followed by division of cytoplasm. तो division of cytoplasm में जो पहली चीज होनी है कि सेल को, साइटोप्लाज्म को दो हिस्से में बंटना है. तो साइटोप्लाज्म को दो हिस्से में बंटने के लिए एक क्लीवेज करो या इनवेजिनेशन. ऐसे ये जो सेल है, अगर ये सेल है और इसके दो हिस्सों में न्यूक्लियर मेम्ब्रेन रीअपियर हो रहा है, एंड यू सी द क्रोमैटिन फाइबर फिर से खुल रहा है, क्रोमोसोम्स खुल चुका है. आई हैव ड्रॉन द टीलोफेज. तो व्हाट विल आई सी हियर चिल्ड्रन? आई विल सी द इनवेजिनेशन इन द nuclear membrane, and the nuclear membrane will you will see an invagination. तो सेंटर में ऐसे हमको एक इनवेजिनेशन नजर आएगी. ठीक है, और ये इन्वेजिनेशन धीरे धीरे डीप होती जाएगी. So this is the beginning of the cytokinesis will begin in the telophase itself. ठीक है, तो हमने क्या देखा टीलोफेज में? हमने देखा कि जो एनाफेज में स्पिंडल फाइबर्स छोटे होने शुरू हो गए, तो उनके शॉर्टनिंग की वजह से द क्रोमैटिड्स गॉट स्प्लिट फ्रॉम द सेंट्रोमियर रीजन, एंड ऑल द क्रोमोसोम्स, द डुप्लीकेटेड क्रोमोसोम अलग अलग हाफ में हैव माइग्रेटेड टुवर्ड्स द पोल्स ऑफ द सेल. फिर उनके चारों तरफ हर वो चीज जो गायब हो चुकी थी इन द प्रोफेज will begin to reappear. The nucleolus will reappear, nuclear membrane will reappear, the endoplasmic reticulum and Golgi body, all the membrane bound organelles, they will all reappear, और chromosomes हम disappear हो जाएगा, वो वापस chromatin fiber के फॉर्म में नजर आना शुरू हो जाएगा, and cytokinesis की शुरुआत हो जाएगी by the appearance of a invagination, एक depression of the nuclear membrane in the center. So next, cytokinesis, जो कि है a cleavage furrow deepens totally in the animal cell and it separates the two daughter cells. तो वो जो डिप्रेशन है, वो अगर मैं ऐसे डिप्रेशन को ज्यादा दिखाऊं तो डिप्रेशन बढ़ता जाएगा. दिस इज द जेनेटिक मटेरियल. ठीक है, this depression or the cleavage or invagination of the plasma membrane will allow for the cell to eventually divide into two by allowing for the division of cytoplasm. ठीक है, the diagram explains clearly, children. Let us read the diagram. The cell membrane is drawn in. Cell membrane अंदर की तरफ चला गया until the cytoplasm is pinched. मतलब वो पूरी तरह से pinch होके अलग हो जाएगा. Pinched into two equal parts. The cytoplasm gets pinched into two equal parts by the invagination or the drawing in of the plasma membrane. And thus, जब वो complete pinching हो जाती है तो दो अलग सेल्स बनके वो तैयार हो जाते हैं. ईच पार्ट कंटेन्स इट्स ओन न्यूक्लियस एंड इट्स ऑर्गेनल्स. हमने देखा कि टीलोफेज में सारे ऑर्गेनल्स आ गए थे, तो आप देखो ये न्यूक्लियोलस आ गया है एंड ऑल द अदर ऑर्गेनल्स विल आल्सो रीअपियर. ठीक है. Next, second point, let us give a reading. The cell membrane starts constricting from the sides and continues inwards until the mother cell is divided into two daughter cells. So that completes the cytokinesis part also in the animal cell. So today, children, I have tried to cover the karyokinesis part of the cell division. What is karyokinesis? Nuclear division, जिसके हमने stages देखे. हमने अपनी सुविधा के लिए stages बोल दिए हैं, जबकि ऐसा कोई stage, ऐसा कोई नाम करके कुछ होता नहीं है when the cell is actually dividing. So for our own understanding, we have said कि पहले जो हो रहा है, पहले जो events हो रहे हैं, we will call them as prophase because pro means early. So early events, what are the early events? We have seen that nucleolus disappear, chromosomes begin to come, nuclear membrane will disappear and all the other membrane bound organelles will also disappear and chromatids हमें दिखने शुरू हो जाएंगे. ठीक है. फिर हमने बोला कि next stage is called the meta. Meta is for center, जहाँ पे हम देखते हैं कि क्रोमोसोम सारे सेंटर पे आ गए हैं और सेंट्रियोल्स जो कि दो पोल्स पे चले गए थे प्रोफेज में, जिनसे स्पिंडल फाइबर धागा निकलना शुरू हो गया है. वो धागे कहाँ जाएंगे? दे विल गो एंड गेट देमसेल्व्स अटैच टू द क्रोमोसोम्स. क्रोमोसोम के सेंट्रोमियर पे वो अटैच कर जाएंगे और फाइनली वो धागे पहले वो बढ़ेंगे फ्रॉम प्रोफेज टू मेटाफेज और फिर वो छोटे हो जाएंगे. तो जब वो छोटे होंगे धागे, जब वो छोटे होंगे तो दे विल ब्रेक द क्रोमैटिड्स इनटू टू पार्ट्स. सो तब वो जाके हमारा क्रोमोसोम्स दो हिस्सों में टूट जाएगा एंड thus we will achieve the division of the nucleus. or the last phase of the karyokinesis telophase में it's almost the reversal of prophase, जिसमें हमने बोला कि जो सारे organelles गायब हो गए हैं, they will all reappear. all the endoplasmic membrane system, that is Golgi apparatus, nucleolus and nuclear membrane, they will all reappear. और chromatids जो थे, that will and chromosomes will disappear and they will once again form into chromatin fiber. ठीक है. उसके बाद साइटोकाइनेसिस शुरू हो जाएगी जब एक इन्वेजिनेशन होगा into the plasma membrane जो कि डीप होता जाएगा. इवेंचुअली साइटोप्लाज्म दो हिस्सों में पिंच ऑफ करके दो अलग डॉटर सेल्स में कन्वर्ट हो जाएगी. So that was about cell division. Hope children you are understanding the cell division and this is only helping you to clarify your concepts even more better and to be able to take up questions nicely and answer them for your. कोई घबराने की जरूरत नहीं है, complicated diagrams को देख के डरने की जरूरत नहीं है. I hope these lectures are clearing all your confusions and fear. So if I'm able to remove the fear of even one child, I feel my effort of putting this on the YouTube is accomplished. So keep on watching my videos. मैं इसके आगे का टॉपिक की वीडियो आपको जल्दी डालूंगी. Thank you very much children and hope you enjoy your day. Thank you.